Okay, so we're now talking about the urinary system. Let me change this to slideshow mode. That's my dog Lyle at the beach in Fort DeSoto. Okay, the urinary system, excuse me, functions to regulate blood volume and the composition of blood. Remember, those hormones are associated with blood volume and blood pressure, and therefore regulates blood pressure. Blood pH contributes to metabolism and excretion of waste products and foreign substances. So remember, when it comes to red blood cell production and then calcium ion absorption, we talked about calcium ion absorption in AMP1, and we talked about it in the endocrine system. And I said, when it comes to the urinary system, we're going to come back again to it. So we're there. So remember, hypoxia stimulates the release of erythropoietin. EPO targets the red bone marrow to increase red blood cell production, then therefore increase oxygen carrying capacity or oxygen content of blood. Parathyroid hormone targets the kidneys to increase calcium absorption from there. It also stimulates the conversion of vitamin D to calcitriol. And then calcitriol then targets the small intestine to increase calcium ion absorption. Okay, organs of the urinary system. We have kidneys, 2 kidneys, about 1.2 million nephrons per kidney. The nephron is the repeating functional unit of the kidney. We have 1 ureter per kidney, or 2 ureters, 1 urinary bladder, and 1 urethra. All right, so here we can see the location of the kidneys. We have the right and left kidney. The right is a little lower than left because of the liver that's found there. We have right renal arteries and veins, left renal arteries and veins. We can see the ureters that transport urine down to the bladder. All right, so the gross anatomy of the kidneys, we have a capsule to the outside, and like any connective tissue, outer layer of an organ, it helps keep the kidneys in place. It helps protect from trauma. It helps to, affirm and connect the blood vessels and the other structures entering, exiting the kidneys. But it also is rather firm, so that it doesn't allow the kidneys to expand too much, because we're going to be really monitoring blood pressure here and adapting to changes in blood pressure. And so we don't want the outer connective tissue to allow the kidneys to expand too much. It wants to be rather firm. Right, we have a cortex in the mid-medullary area, just like the adrenal glands. The cortex is the most superficial region. It contains most of the nephron. The medullary area is deeper to it and contains the renal pyramids. And within these renal pyramids, it's a darker staining region. We have the collecting ducts in the loops of Henle. And then from there, the fluid that we're modifying, called filtrate, leaves that region as urine. It gets passed into what's called major and minor calyces, which are little tubes, so to speak, transporting urine to a larger region called the pelvis of each kidney. And then from there, we have ureters that will then transport urine down to the bladder. So here, if we're looking at the pictures of the kidneys, the cortex is the lighter pink staining region. And we have little indented finger-like projections of cortex called the renal columns that extend toward what's called the renal pelvis. And in doing so, it kind of divides the medullary area up into pyramids or pyramid-shaped regions. So those are the renal pyramids, of the darker staining, kind of triangular portions. It contains medullary tissue. And when we look at the nephrons, parts of the nephrons are going to be found in the cortex, parts in the medulla. At the tips of the medulla, what's called the papillary regions, is where urine will drip out as it's produced, and it travels into these minor and major calyces, they're called, to the renal pelvis and out in the ureter. We'll also look at a certain amount of the blood circulation through there as well. Okay, so the nephron is the repeating functional unit of the kidney, about 1.2 million per kidney. It is composed of one major region called the renal corpuscle. The renal corpuscle has two kind of structural components to it. The capillaries, the blood supply within the center, and then the capsule part to the outside. Together, those two structures form the renal corpuscle. Then from the renal corpuscle, we have the tubular system, which includes the proximal convoluted tubule, the descending ascending loops of Henle, and then the distal convoluted tubule, and then finally the collecting duct. And that's what then is formed by, oopsie doodle, multiple distal convoluted tubules of different nephrons kind of merge to form a collecting duct, and then multiple collecting ducts are going to transport urine into the calyces, ultimately to the pelvis and onto the bladder. So here we're looking at kind of in the middle to the left-hand side, we see one great big renal corpuscle. It often looks like a ball. We have red vessels entering and exiting. So we have an overlap of the cardiovascular system here, the red and the blue vessels, and we have the nephron, which is composed of the renal corpuscle, and big black letters there, the renal corpuscle. You have the capsule to the outside and the capillary network inside called the glomerulus or the glomerular capillaries. We will filter blood here and it hits the renal corpuscle, the capsule wall, and then forces whatever gets through that filter and fluid into the proximal convoluted tubule. So convoluted means it's kinked on itself. It's a hose area that's kinked on itself. So here we have the proximal convoluted tubule showing you the arrows that the fluid will travel from the proximal convoluted tubule of the PCT. It travels into the descending loop of Henle, goes around a hairpin turn, travels up the ascending loop of Henle to the distal convoluted tubule, and then multiple distal convoluted tubules will merge into a collecting duct and then transport the fluid out into the urinary pathway. So the fluid within this system is called filtrate. It's similar to when we have fluid that enters into the lymphatic capillary and then onto lymphatic vessels. We call the fluid in there lymph. Well, the fluid within the nephron is called filtrate. We can see the major blood supply. We have arteries transporting blood to the nephron. The smallest vessel that's entering that nephron is called the afferent arteriole. So arteriole capable of vasoconstriction dilation. It branches off into capillaries within that renal corpuscle and leaves as the efferent arteriole. So we're going to look at that as well, and identify them specifically. There are other branching capillary networks from that efferent arteriole, but this is a good picture to show the direction of blood flow, to that renal corpuscle and back out. And then all the venous blood will eventually merge and form renal veins and to the inferior vena cava and back to the right atrium. What is good about this is over to the right-hand side, it talks about the endothelial changes, along this pathway. So we start at the capsule, the glomerular capsule, is rather a thin layer of cells. The capillaries to the inside, capillaries are simple squamous epithelium. And this is where blood will be filtered. Whatever can get through the walls of that squamous capillary, just like at any capillary bed, whatever can get through will leave because of the hydrostatic pressure. And it kind of hits the walls of that capsule and then is forced down the rest of the tube system to the PCT, proximal convoluted tubule, distal, descending loop of Henle, ascending loop of Henle, so forth and so on. The proximal convoluted tubule, is lined by cells with microvilli that helps increase absorption. And it also slows down the movement through there a little bit so that you can absorb as much as possible of certain molecules that we want to absorb there, and we'll go through that. Then when we enter the distal convoluted tubule, we're lined with, parts of the loops of Henle. We are going to be lined with a lot of squamous cells. Right? The squamous cells will be the thin portions of a loop of Henle aligned with simple squamous epithelium. When we go to the thicker portions, we're lined with either simple cuboidal epithelial tissue or simple columnar. But because the cells change, so do some of their functions. Like the distal convoluted tubules are generally composed of columnar cells, and so are the collecting ducts. And then urine leaves. Okay, so we are going to go to the next slide that is talking about not only the nerve supply, but now the cardiovascular supply and pathway, because everything has a forward direction pathway into and out of all these structures. Right, well, the renal nerves, sympathetic neurons regulate blood flow. Remember that sympathetic nervous system is in control of, vasoconstriction and dilation. All right, so it's regulating the blood flow, the amount of blood circulating into the kidneys at any point in time. All right, so the pathway, we have renal arteries transporting oxygenated blood to the kidneys. They branch off multiple times into smaller arteries until finally we reach the tube or the vessel that's going to enter that renal corpuscle. And it does so via the afferent arteriole. So remember, arterioles are capable of vasoconstriction dilation. And so they transport blood into the glomerulus, rather large diameter compared to the efferent arteriole. Then that efferent arteriole branches off into the glomerular capillaries or the glomerulus, composed of fenestrated capillary network within the glomerulus. So just like any capillaries, the blood is entering under force. So molecules that are capable of passing through the little walls between the squamous cells do so. Anything to get through is going to get through. It's going to hit the walls of the capsule and move down into the proximal convoluted tubule. The molecules that can't leave are going to remain in the capillary bed and travel to the other side and leave glomerulus in the efferent arteriole. Right, so it transports filtered blood out of the glomerulus, and it is a narrower diameter compared to the afferent arteriole. Why is that important? Keep this in mind when we're identifying these same structures on models and things in lab, is that both of these are red vessels. And unless you see really where the vessel is coming from and where it's going to, in the case of an efferent arteriole, there's no color difference, but the diameter is different. The afferent arteriole has a larger diameter than the efferent. So the one entering the afferent arteriole is accommodating more blood volume as it enters the glomerulus. But because we're filtering the blood and a lot of water and molecules are going to leave the blood, not necessarily get reabsorbed, although there is a certain amount of osmotic pressure to draw, molecules back in. Most of it, because now it has a direct pathway into the proximal convoluted tubule, is going to go in that direction. So the blood leaving the renal corpuscle, is a lower volume. So the efferent arteriole doesn't have to be as large or as wide, because less blood and fluid is going to have to be transported through that vessel. So it's a narrower diameter. All right, peritubular capillaries. From there, the efferent arteriole branches off into two capillary beds, one that stays in the cortex region and surrounds the structures found there, like the proximal convoluted tubules and the distal convoluted tubule. They're called the peritubular capillaries. Supplies the PCT and the DCT where absorption and secretion occur. The other branch travels down into the medulla that will surround the loops of Henle, and adjacent to significant portions of the collecting duct. They go in red, because they're rich in oxygen, but we have a lot of gas exchange there and, exchange of nutrients to a certain point as well. and they will leave as what's called the blue vasa recta. So it branches off into this very unique shape of capillary bed in the medullary region. It is all called the vasa recta, but you'll see some portions of it that are red, but we have a good amount of gas exchange there, so it's going to leave more like a vein, transporting venous blood, richer in carbon dioxide. And then all the vasa rectas ultimately merge to form larger veins until they become a renal vein, a right or left renal vein, transporting deoxygenated blood, low waste level blood into the inferior vena cava. All right, so here we see sort of an overlap of the blood vessels and these renal, the nephrons. You see that artery and vein that's running on the horizontal? That's to tell you this is the junction between the cortex above it and the medulla below it. All right, now here we have on the right and left, each a nephron. And sometimes more of the nephron resides more in the cortex and less in the medulla, where you see the little loops of Henle that extend in the medulla. And then a lot of them are just like the one to the right. You have portions of, of the nephron in the cortex, but significant portions in the medulla as well. All right, so, it's a difference between being called more of a cortical nephron versus a juxtamedullary nephron. But in any event, we're going to look at these. So we can see the blood supply. So let's look at the big red there at the cortex medullary junction. The blood is shows you the direction of blood transport upward in that big red vessel. We're going to have a branch that goes to the nephron to the right-hand side. It enters that renal corpuscle as the afferent arteriole branches out into a capillary bed or glomerular capillaries and leaves as the efferent arteriole. And you can follow that pathway of blood on the nephron on the right and on the left. Now, the one on the left gives you a good indication of how these are going to branch off. So when we look at the efferent arteriole of the nephron to the left-hand side, you can see that it's going to branch off into a peritubular capillary network and a vasa recta capillary network. They all ultimately have a certain amount of gas exchange and reform to form a vein, which ultimately becomes the renal vein to the right and the left. So we can see blood entering, blood exiting, and then traveling in the blue venous cardiovascular pathway. Okay, so when we're looking at slides, we are either going to be looking at either generally, the cortex region or the medullary region. So once you know what parts of a nephron are primarily found in the cortex versus the medulla, then identifying them on slides is pretty easy. So here we have to the left-hand side, we're looking at the cortex, and it's showing you essentially down below that colored region on that kidney where this tissue is coming from. And then over to the left-hand side, to the right-hand side, it's dark stained the medullary region and what a slide of it might look like. All right, so for all intents and purposes, in the cortex, we find the glomerulus, and we find lots of PCTs and DCTs. So when we're looking at this slide here, you see how you see those big nubby balls there? That is the glomerulus. It is composed of the capsule, right? The BOWMANS capsule or glomerular capsule to the outer region. And it's kind of dark staining. And then within it, look at all those red blood cells in there. That is the glomerulus or the glomerular capillaries. In this particular picture, you can see the blood supply into and out of it going right through the center. So I see 3 renal corpuscles here. So if you can see those knobby-like structures, you know you're in the cortex, because that could be the question. What region, quote unquote, of the, or maybe what organ is this? First, is the kidney. Say, what region are we in? This would be the cortex. Over on the left-hand side, what region? It would be the medulla. How do I know the left-hand side is the cortex? Well, I can see these renal corpuscles, so you know you're in the cortex. And you see some of these little round tubes on end. That is a simple cuboidal epithelial tissue of the PCTs and the DCTs. You know, in AMP1, when we looked at tissues, you know, we looked at some of the tissues in the integumentary system, because we often had a separate slide for just simple cuboidal epithelial tissue. You know, and if you were in my class, I told you, I was like, these generally come from the kidneys. Now we're right back here. That's what we're looking at, simple cuboidal epithelial tissue, right? Single layer cuboidal cells. Wilma Flintstone's necklace, we used to call it. Okay, and so not that you have to distinguish between a PCT or a DCT here, but you know, that's generally what you're looking at here. Okay, over to the left-hand side, then you go, okay, well, what do I generally uniquely kind of find in the medulla? We find our descending and ascending loops of Henle and collecting ducts. And that's what you're looking at here. And so you know you must be in the medulla because you don't have any renal corpuscles there. So I hope that helps. Okay, this is a fantastic picture I scarfed from a website. Now here we have an efferent arteriole, you know, mostly PCT that you're looking at, one DCT here, and a lot of erythrocytes. So when you can magnify it well and look at it, it's really pretty fascinating. And so this is our repeating functional unit of the kidney. About 1.2 million, they're packed all in there. Each nephron has its own blood supply into and out of it, and therefore filtering your blood, having an effect on all the different things we mentioned functionally: blood pressure, blood volume, blood pH, getting rid of waste materials. You can see to the right and to the left. All right, so let's look at that. And this is kind of interesting to see. You see on the bottom at about 5 o'clock, the afferent arteriole entering into this glomerulus, because you can see all those erythrocytes there. You can see to the above, you know, above that, you see that single tubule, that's a proximal convoluted tubule. You see how it looks kind of furry in the interior? That's the cilia of the cuboidal cells there. So that's one way of recognizing technically whether you are looking at a proximal convoluted tubule or a distal convoluted tubule. To the other side of that afferent arteriole is a distal convoluted tubule, and it's forming what's called the juxtaglomerular apparatus. Now, do you see that row of cuboidal cells? See how it doesn't really look furry in the center? That's because it's not. That's a distal convoluted tubule. And that's where we find what's called the juxtaglomerular apparatus. Just FYI, if you want to know what you were looking at there. All right, when it comes to urine formation, it requires these 3 phases or processes: glomerular filtration, tubular reabsorption, tubular secretion. So in glomerular filtration, filtrate includes everything potentially found in blood except formed elements and plasma proteins. So what we're saying is glomerular filtration is going to occur one place and one place only along this system. All right. Things either get through those capillary walls of this capillary, the squamous cells that form the capillary, or they stay inside the blood. And things that should never get through those capillary walls and become part of filtrate through the rest of the nephron, the tube system of that nephron, should be things that we don't want to get rid of, right? Things that are too big. Not all the water is going to get through either. You know, some of the water is going to, and fluids are going to, plasma is going to stay in the cardiovascular system. But things that should never get through the filter are your formed elements, red blood cells, white blood cells, platelets, and plasma proteins like albumin. Right? So if they are found in urine later, you know, abnormally, then you know something's not right. All right, we can either reabsorb material, which are substances moved from the renal tubules into the interstitial fluid, where then they diffuse into the blood. So even though some material gets through that filter, or a lot of molecules get through that filter, we don't necessarily want to throw them out, because the filter is designed to just separate things by size. not by whether we really want to get rid of them or not. So once we produce the filtrate in the proximal convoluted tubule, gives us an opportunity to take back and reabsorb molecules we don't want to get rid of. So there'll be certain ones we maximally reabsorb in the proximal convoluted tubule, and then elsewhere down that tube system, loops of Henle, distal convoluted tubule and collecting duct, we will have unique opportunities to absorb certain things and secrete other things along that pathway as well. But all we're saying is reabsorption is taking molecules from that are now within that nephron as filtrate and putting it back in the blood. In the same respect, because we'll have capillary beds like peritubular capillaries and vasa recta surrounding these tubules, depending whether the part of the nephron is in the cortex or the medulla, we can actually get rid of some extra molecules too, because not everything will get through that filter. So as this filtrate passes along the regions of the nephron, from the glomerulus or the renal corpuscle to the proximal convoluted tubule, descending loop of Henle, ascending loop of Henle, distal convoluted tubule, and so forth, we will have opportunities to reabsorb or secrete. So we start looking at those. This just shows you where the three processes will occur and the direction of molecules. Right, so here we have blood entering into that renal corpuscle via the afferent arteriole, branches off into a capillary network. Hydrostatic pressure behind it pushes molecules and fluid to the wall of the capsule, glomerular capsule or the BOWMANS capsule. Most of us still refer to it as that. And then that material passes into the nephron tubular system. Well, reabsorption or tubular resorption is two. It goes from inside that tubule into the blood. And tubular secretion means we're going to take some of the molecules from the blood into the tube system. All right, so the BOWMANS capsule is the cup-shaped portion of the radial tubule that surrounds the glomerular capillaries. It collects the filtrate. That fluid is going to hit the walls as it leaves capillary network due to hydrostatic pressure, hits the BOWMANS capsule, and it gets transported, into the proximal convoluted tubule. So the glomerular capillaries within it are otherwise referred to as the glomerulus. This is where filtration occurs. The ball of capillaries that allow solute-rich filtrate to leave blood and enter the tube system as filtrate. Capillary walls are fenestrated. All capillary walls really are fenestrated. They contain small pores that allow the passage of small solutes. Remember, hydrostatic pressure pushes molecules out, and then the kind of hypertonic nature of blood as it passes from one side of the capillary to the other kind of draws some of that fluid back in. And also partially because it hits the walls of the BOWMANS capsule and it kind of pushes back in as well a little bit, but most of it, will not be drawn back in, just like what happens in a regular capillary bed. That's why we have the lymphatic system to capture that interstitial fluid. Similar thing happens here. But instead of the lymphatic system capturing that fluid, the fluid would be forced down through the other regions of the nephron. All right, so what may be found in filtrate, at the beginning at least, it's normal to have water, glucose, amino acids, sodium, potassium, urea, bicarbonate ions. Anything that is potentially being transported in blood can pass through, and as long as they're small enough, can pass through the walls of that capillary and become the filtrate entering into the proximal convoluted tubule and so forth. What should not be normally found there are red blood cells, white blood cells, platelets, formed elements, and albumin. This just shows you, here we have the renal corpuscle. You can see the BOWMANS capsule to the outside, the capillaries to the inside, and the hydrostatic pressure pushes fluid out that will then be transported down through the tubular system of the nephron. Okay, so now we enter into the proximal convoluted tubule. Very long kinked hose, kind of like a small intestine, with villi on those cuboidal cells. That is designed to facilitate reabsorption. Most reabsorption of molecules is going to occur in the proximal convoluted tubule, just like most, 99% of the absorption of nutrients and water is going to be in the small intestine. All right? If they are of normal levels, we will absorb or attempt to absorb 100% of the glucose that became filtrate and now is in the PCT. 100% of the amino acids, because we never want to get rid of amino acids because those are the building blocks of proteins, and we have tons of proteins in our body that need to be made all the time. Albumin, antibodies, all kinds of things, clotting factors. And glucose is the preferred molecule of metabolism in cells. So even though they're small enough to get through, we don't want to pee it away, so to speak. So we will, in the proximal convoluted tubule, reabsorb 100% of those, as long as they're normal levels. About 85% of the bicarbonate that's there, and a lot of sodium. Things that we might secrete is ammonia. I think I said before when we were talking about metabolism is that we never reabsorb ammonia. And we will often secrete hydrogen ions. So right at the beginning of this system, it gives us a chance to get rid of some of the excess hydrogen ions, and therefore trying to regulate pH. Now we enter the loops of Henle. We have the descending loop of Henle, then the ascending loop of Henle. So now that we have filtered passing through the descending loop, it is composed of simple squamous epithelium. And that region is permeable to water only. So the descending limb is permeable to water. Water is reabsorbed and drawn into the interstitial space and into the blood supply there. So we reabsorb water in the descending loop of Henle. The ascending loop of Henle changes epithelium to a simple cuboidal epithelium. And now we are no longer permeable to water, but we do reabsorb sodium, potassium, and chloride. Here we can also secrete a little more urea. Remember, urea is kind of a modified ammonia. Right, so we reabsorb water on the downward descending loop. We reabsorb sodium, potassium, and chloride ions on the ascending loop. Okay, then we get to the distal convoluted tubule and collecting duct. So for all intents and purposes, we kind of put these together because there's, depending on, whether you're at the beginning of the distal convoluted tubule or the end of the distal convoluted tubule, altogether, it pretty much mimics the function of the collecting ducts. And there's only one small exception, and that's where the portion, arguably of either, the distal end of the ascending loop of Henle or the proximal end of the distal convoluted tubule, depending on the textbook you use, will tell you that that is the region, that forms the juxtaglomerular apparatus. But functionally, everything else, we're going to put together. That if it happens in the DCT, it's happening in the collecting duct as well. All right, so it's composed of simple cuboidal epithelial tissue. Contains receptors for antidiuretic aldosterone, parathyroid hormone, and calcium pumps. Okay, so low blood levels of sodium and low blood volume. Remember, so we said low blood volume, low blood pressure, maybe increased osmolality, of blood will stimulate the release of aldosterone to increase our sodium chloride absorption and secrete potassium. So remember, aldosterone actively pumps sodium, chloride follows, where salt goes, water goes. And so we are essentially pumping salt back into the blood and drawing water with it. And at a cost of absorbing the sodium, we must secrete potassium. When we have low blood volume or pressure, we can secrete antidiuretic from the posterior pituitary. Remember, that's going to stimulate just the reabsorption of water. Just the reabsorption of water. And then if we have just by itself low blood calcium levels, Parathyroid hormone will be released, and that stimulates reabsorption of calcium, from the filtrate, so we don't just pass it off into urine and eliminate it. Filtrate is called urine only after it leaves the collecting ducts. So once the fluid technically is entering into what's called the calyces, that are capturing that fluid and funneling it down to the renal pelvis, that then and there, that's technically where you start calling it urine instead of filtrate. Urine drains into the calyces, the renal pelvis, into the ureters. So what we're saying here is when it comes to these hormones, antidiuretic, aldosterone, parathyroid hormone, it's going to stimulate the same reaction in both the DCTs and the collecting ducts, to modify blood volume and blood pressure. Maybe in the case of being hyperkalemic, if you have too much potassium, right, you can use aldosterone so that you reabsorb sodium to secrete potassium. Remember, the parathyroid hormone, has nothing to do with blood volume or blood pressure, but it's trying to address a low blood calcium level. So it's going to target these structures as well. When it comes to the end of this system, we're going to be, primarily having certain functions that we're focusing on to address. All right. And I'm going to talk about that when we get through kind of this chart. This chart helps you kind of put together the different nephron parts. So over on the left-hand side, we have glomerular capillaries of the renal corpuscle, or PCT, or descending loop of Henle, ascending loop of Henle, and then together the DCT and the collecting duct. Then we have next to it and on the the horizontal axis, I beg your pardon, the processes of urine formation, filtration, reabsorption, and secretion. So we're going to look at, hey, where does filtration occur? Where does reabsorption and secretion occur? What molecules, are being reabsorbed or secreted along, in that particular area of the nephron? So up on the top, this is as a reminder, found in filtrate, at least at the beginning, maybe not at the end of becoming urine, but found in filtrate, is really anything that's being transported in plasma, right? Water, glucose, amino acids, sodium, potassium, urea, bicarbonate, all kinds of hydrogen ions, all kinds of material, different hormones, right? That's why we use urine to test, whether, at early stages of pregnancy, to see if human chorionic gonadotropin is in there. What should not be found normally there are red blood cells, white blood cells, platelets, are formed elements, and albumin. All right, so if we start at the renal corpuscle, this is where filtration occurs up at the top, right? Everything except protein and formed elements. Right? So anything else that's there can potentially pass through that wall of the capillary and become filtrate and passing into the proximal convoluted tubule. There is no reabsorption technique or secretion occurring there. But then as we enter into PCT, what might be reabsorbed? Everything in there. Water, sodium, potassium, 65% of that. Glucose and amino acids, about 100% of that. And about 80 to 90% of the bicarbonate that's there. What can be secreted? Hydrogen ions, ammonia, some drugs and hormones. All right, then we go to the descending loop of Henle. Permeable to water only, reabsorption of water. Travels down the descending loop of Henle, makes the hairpin turn, starts ascending as the ascending loop of Henle, and now we change to cuboidal cells, and thus we are no longer permeable to water, but we are permeable to reabsorb sodium and chloride and potassium. Then we enter into the distal and convoluted tubules. And now the last things we want to do, keep in mind, are going to be, balance blood volume, blood pressure, using the hormones, of aldosterone and antidiuretic. We can try to reabsorb calcium because of parathyroid hormone. And then we're going to make adjustments to pH. The blood that's passing through those capillaries, right now we can do some final tweaking to the pH there. If we have a very low pH of blood, we can secrete hydrogen ions and reabsorb bicarbonate. If the pH of the blood is higher than what we want, then we'll do the opposite. We can reabsorb some hydrogen and secrete bicarbonate ions to decrease the pH. Now, I'll tell you that technically we don't really reabsorb hydrogen ions per se, but we're going to address it that way. If the pH is low, how do you elevate the pH? You get rid of hydrogen ions and you reabsorb as much bicarbonate so it can interact, like carbonic acid cycle, interact with hydrogen ions to reform carbonic acid and then reform maybe carbon dioxide and exhale. Okay, if it's higher than what you want, right, then you want to decrease the pH. Thus, you want to increase the hydrogen ion concentration and get rid of bicarbonate into urine. So that's why it says down there, reabsorption bicarbonate or hydrogen ions as needed to buffer the blood, 7.35 to 7.45 normal pH, or we may secrete a bicarbonate or hydrogen ion as needed to buffer the blood. So hydrogen and bicarbonate ions are going to be going in opposite directions. Okay, urine transport, filtrate from the convoluted tubule, in the papillary duct, drip into a minor calyx. Minor calyxes merge to form major calyces, and they merge to form the renal pelvis. Collects urine, transports to the ureters. This fluid, once it passes from the nephron into a minor calyx, we refer to as urine. Okay, the ureters, microanatomy of the ureters, who have three layers. We have a mucosa, muscularis, and adventitia. The mucosa is composed of transitional epithelium. The muscularis layer, we have two layers of smooth muscle. We have the outer connective tissue layer, same as any other outer connective tissue layer. Anchors the ureters, anchors blood vessels and nerves, to these structures. It has a very unique presentation on a histological slide. It kind of looks like a sand dollar. Generally, when you're looking at a slide of a ureter, you can see the whole thing, like an artery on end. The bladder is too big. You'd have to have kind of a pie wedge to it. So it's the difference of looking, at like a one artery on end versus maybe when we looked at the stomach, you get kind of a pie wedge of the stomach, or even of the small intestine. They look so similar. Okay, function of the ureters: transport urine from the pelvis to the bladder. Distension of the ureter created by incoming urine stimulates the contraction of the muscle. So once it starts to stretch, it's kind of like a recoil mechanism. Once it stretches, it stimulates it to contract and propel the urine down toward the bladder. Thus, it has, that peristaltic effect of inner circular outer longitudinal muscle that propels it forward. Crystallization of calcium can form renal calculi, or what's called urinary stones, and they may block ureters. Very painful process, very painful. Here is that histological slide of a ureter, so that you usually can see it just like this in its entirety. And you see, it kind of looks like a sand dollar on the inside. The first layer adjacent to the lumen, which is, this transitional epithelium that we find there in the muscle, it can stretch like blowing up a balloon, like a big blood vessel, but it will collapse back down when it's not propelling urine. We don't want to have a lot of air in there. Right? So the first layer is mucosa, composed of transitional epithelium. We have two layers of muscle, right? Circular and longitudinal. And then we have the outer connective tissue, which is the adventitia. Right, then we go into the urinary bladder. Location is posterior to the pubic symphysis, anterior to the rectum in males, and the vagina and rectum in females. Histologically, it's very similar to a ureter, except the muscular layer we call the detrusor muscle has its own name, and it has three layers, kind of like the stomach, instead of the two that we look at in the esophagus and the small intestine. Right, so very similar. Then we move on to the urethra. Here, the mucosa changes from transitional to pseudostratified columnar, believe it or not, and then ultimately to stratified squamous epithelium. at the distal end of the urethra. The female's urethra is short, and thus why females are a little more inclined to have to be, I guess, susceptible to urinary tract infections, because bacteria can travel up the urethra and take residence in the bladder and further travel up the ureters into the kidneys and become even more invasive kind of infection. The male's urethra is quite a bit longer, and that, once it passes the prostate gland, the male urethra is capable of carrying urine or transporting urine or semen. Micturition is the act of voiding urine. Urine leaves the urinary bladder by micturition or urination reflex. Parasympathetic nervous system coordinates that detrusor muscle to contract, followed by internal and external sphincter relaxation. So micturition, is and voiding of the bladder is a parasympathetic function. That's why in the autonomic nervous system in AMP1, I said, we had this little mnemonic kind of, well, not really mnemonic, but saying for an acronym for the systems that are elevated in parasympathetic stimulation. We said it was SLUD. The systems that increase are salivation, lacrimation, urination, digestion, defecation. Well, it's the parasympathetic division that stimulates the detrusor muscle to contract, and that's followed by coordinated relaxation of the two sphincters. You have the internal sphincter that is at the junction between the bladder and the urethra, and the external sphincter would be at the distal end between the urethra and the outside environment. The internal sphincter is smooth muscle, thus is controlled by the autonomic nervous system. But the external sphincter is skeletal muscle, so it's under volitional control. Here we're looking at a bladder. We can see two ureters to the top, traveling kind of to the posterior aspect, and they merge and connect to the bladder on the inferior aspect, not at the top, but at the bottom. The trione bone is the triangular region between the openings of the two ureters and the urethra. That's that triangular area. You have an internal urethra and an external urethra. Internal urethra is smooth muscle, external urethra is skeletal muscle. You can see it to the sides. Okay, so that's where we're going to break. Give you a chance to kind of digest this. After this, we're moving on to a lot of the physiology that occurs in the kidney. We already talked about some of that when we talked about the filtration, reabsorption, and secretion of different molecules. But after this, we take it kind of to another level. We talk about some of the hormones again, what's reabsorbed and secreted. We talk about the different components that can be normally and abnormally found in urine for urinalysis. And we talk about what else? Some other aspect of physiology. Glomerular filtration rate, and what that's about. Okay, so we're going to break here and then resume with urinary system 2 Zoom.